the format for the test one, usually it's going to be on the structure. Okay, structure. Means that it is not objective. It is subjective. Subjective. Okay. So the subjective, and then essay most probably. Okay. Yeah, I haven't do the question yet, but most probably the questions will be subjective and essay. All right. Okay, so last lecture we have covered on the process of transamination. Okay, so how the transamination takes place, and there are also a few examples given to you. Okay, about how the process of transamination takes place in different amino acids. So let's say previously we have discussed on how the alanine, all right, how the aspartate go through this process of transamination. Okay, so and remember each of the amino acids. Okay, each of the amino acids, for example, here aspartate. Okay, so we're going to get the product which is the oxaloacetate. This is a form of alpha keto acid. Okay, alpha keto acid. So alpha keto acid for different amino acids is different. Okay, so it is accordingly to what type of amino acids that involved. So let's say for the alanine, so what is the alpha keto acid for alanine? So we have here the alpha keto acid for aspartate is oxaloacetate. How about the alanine? Okay, pyruvate. All right. Okay, so pyruvate. And the same thing goes to another amino acid. So they have a different name or different types of alpha keto acid, which is for the transamination of that particular amino acid. All right. Okay. Okay. So as we have discussed before, the concentration of the glutamate is going to get increased. Okay, it's going to increase because each of the amino acid is going to be degraded, which is they are going to go through this transamination process. So during this transamination process, the amino groups of this amino acid is going to be removed. We get the product as the alpha keto acid plus the glutamate. All right. So we can see from every amino acid, which is we have 20 amino acids, right? So 19 because we have already glutamate there. Okay, so if let's say 19 amino acids is going to go through these transaminations, then we're going to have a bunch or high concentration of glutamate. All right. So what's next? What's next when we have these glutamate, such a high concentration of glutamate after the transamination process? Okay, so so the first thing is that this is very important, okay, because these glutamates will become as the donor for the biosynthetic pathway. So this is going to be a part of the source of the amino group, okay, for the biosynthetic pathway. And also the amino group from the glutamate will be excreted. It will be excreted. So we're going to see later, okay, what are the pathway, what are the processes involved for the excretion of the amino group. Okay. All right. So the process of deaminations that we have discussed before, which is go through the transamination process. So as we have discussed, we have the enzymes. Here we have the enzyme that transaminase that catalyze the process of transamination, and we have the product, which is a compound that we call as alpha-ketoglutarate, right? And the product is also alpha-keto acids, which form from the amino acid and the glutamate. Okay, so this is the alpha-keto acids and glutamate, which is a product from the transamination. Okay, and okay, okay. So then the amino acids, which is that has been collected as the glutamate. Okay, so this amino group from glutamate is then need to be removed, right? Because we have a very high concentration of glutamate, and then obviously we need to remove the part of the amino group from the glutamate itself. In the next step, and the next step is the next step where we have to remove the amino group from the glutamate is what we call as oxidative deamination, or which involve the glutamate dehydrogenase. So this is the next step. Okay, the next step after transamination process. And okay, the first the main the first characteristic or the first properties of this oxidative deamination process is that it will reforms the alpha-ketoglutarate. Okay, it will reforms the alpha-ketoglutarate with the other product, what we call as the ammonia. Okay, so as you can see here, okay, from the transaminations, we have the first amino acids. Okay, we have the amino acid, which is the first substrate, and then we have the alpha-ketoglutarate, which is the second substrate in the transamination, and we get the product, okay, which is the alpha-keto acid and the glutamate. But then during the next step of the process, which is what we call as oxidative deamination, okay, the oxidative deamination, during the oxidative deamination, we actually reforms, okay, where we get back the alpha-ketoglutarate, right? So here is how the process takes place. So we have the glutamate, and then of course in the presence of the water, okay, the water, and then in the presence of NAD and NADH as the coenzymes, right? And of course we need the enzymes. Okay, we need the enzymes, which is glutamate dehydrogenase in this case. And the products is going to be the ammonia plus the alpha-ketoglutarate. So this is where the reformation, okay, where we reformed back the formation of this alpha-ketoglutarate during this process of oxidative deamination. Okay. Right. So so in this case, when we talk about the oxidative deamination, this is this step where we want to remove the amino group from the glutamate itself. Okay, so because we already have a bunch of glutamate in high concentration, okay, in high concentration. So what's next is that we have to remove the amino group from the glutamate. So this is a process of oxidative deaminations. So what we get after we remove this amino group from the glutamate, later it will become the ammonia. Okay, it will become the ammonia because in the transaminations, the amino groups is attached to the alpha-keto acid, which becomes the whatever product for that particular amino acid, right? But then in this case, in the oxidative deamination, where the products, where the substrate is the glutamate, the amino acids, the amino groups is going to be converted into this a form of ammonia, okay, NH4. So this is the difference between the transaminations, what happened in the amino acid in transamination, and the one that's involved in oxidative deamination, right? And as I said, in this process, oxidative deamination, we will going to reform the alpha-ketoglutarate, right? And okay, so the location where the process of oxidative deamination is the liver and kidney, okay, and the exact location is the mitochondria, okay, the mitochondria. of course, the mitochondria in the cell of liver and kidney. Yeah? Okay, so it happened in the presence of glutamate dehydrogenase. So this is the enzymes that responsible for the process of to convert the glutamate into the alpha ketoglutarate and also the ammonia. Okay. Okay. So in this case, in the process of oxidative deamination, so there are a certain requirements. Okay, certain requirements that needed for oxidative deamination. So the first one is that if, let's say, there is a presence of the GTP in the cell, so there will be no such conversions of glutamate into alpha ketoglutarate and ammonia. Okay, so this process is going to be inhibited in the presence of high concentration of the GTP. So that is the limitation of this process. Okay, and then if, let's say, there is the presence of the ADP, okay, ADP, then this process is going to be resumed. Okay, dia akan berlaku, proses ni akan berlaku conversion of glutamate into alpha ketoglutarate plus ammonia if in the presence of the ADP. Okay, and of course, as I said, we need the NAD or NADP plus, and it will be there will be reduction process of NADP or NAD plus to the NADP or NADPH. Okay, so this is the where the how the process takes place in the oxidative deamination. Okay, right. As I said, it will be inhibited by ATP or GTP. Okay, GTP or ATP. Okay, and it will be activated in the presence of ADP or GTP. Okay, and of course, it used the NAD plus as the cofactor in this case, but not PLP. So where does the PLP works or takes place? What process PLP involved? Class, in what process the PLP involved? Okay, alright, so strong transaminations. Okay, so jangan babak lupa pula. Alright, so transaminations. So PLP will be the cofactor for the transaminations, but in oxidative deaminations, the cofactor is NAD plus or NADP plus. Okay, so there is two lah. Okay, another one is NADP plus, and so the inhibitor. Okay, the inhibitor for this process is the ATP and GTP, and it will be activated in the presence of ADP and GTP. Okay. So one thing that I would like to ask you here, why do you think that in the presence of ATP and GTP, so this process of oxidative deamination will put on hold?
ATP and GTP.
Okay, why is that? Why does such things happen, such inhibition takes place when there is a high concentration of ATP and GTP? But in case of there is a high concentration or the presence of ADP and GTP, the process of oxidative deamination will continue again. So why such things happen? Okay, so then we said high concentration of ATP and GTP, which is equal of high energy. So then what? So why does it stop? Why does it cost a while? Well, in the presence of high energy. Mhm. Does not need alpha ketoglutarate. Okay, then what? How can you relate to um, how can you relate this ATP, alpha ketoglutarate, high energy? Uh, yang ni kena buat essay lah kan? Okay, okay, tak adalah kena buat essay. Yeah, so I need you to understand the what is actually happening here. What is actually happening? Why does apa kaitannya? What is the relationship? What is the relationship between this this inhibition, these activations, and then we have these enzymes that provide the alpha ketoglutarate to the TCA or citric acid cycle. So all this actually it's actually relate to each other. So I want you to find out about this. Okay. So all right. And okay, so this is how the inhibition, okay, which is the by GTP and activated by the ADP or GDP. Okay, so there are two forms of the energy, which is GTP and the ATP, which is a very high energy, very uh, um, very high energy lah. Okay, GTP dengan ATP is in a high form of energy, and we have also ADP and also GDP. Okay, so that is a lower form of the energy. Okay, so this this is a regulation of the GDH or glutamate dehydrogenase. So what is needed for this glutamate dehydrogenase to work to catalyze the process of oxidative deamination of the glutamate into the alpha ketoglutarate and also the ammonia. Okay, and so when the energy supplies are low, so this enzyme provides the alpha ketoglutarate to the TCA. Yeah, so here the process that we are talking about right now, which is oxidative deamination, is actually linked. So is actually linked to the process of citric acid cycle. So the product that's produced in this process, this is actually the product that will be channeled into the TCA. Okay. All right. So the summary of the amino removal. Okay, amino removal. So starting from the transamination, antioxidative deamination. So this is the summary of the amino group or amino group of the amino acids removal. Okay, so we have the amino acids. Okay, so when we combine the transamination part with the oxidative deamination, so this is what we get. Okay, what we get starting from the this first substrate, which is the amino acids, and then we have the ketoglutarate as the second substrate, and then transamination takes place, and we get the keto acids, and then the glutamate as the product. Then glutamate then will be the product, will be the substrate for the oxidative deamination. All right. So they need the other cofactor, which is the NADH, and also the ADP, right? ADP or GDP as the to give it a signal for oxidative deamination takes place, and we have the and then we have the products for oxidative deamination, which is back to alpha ketoglutarate and then the ammonia. So this is the summary, okay, the total summary of what's happening to this amino group of the amino acids. Okay, starting from transamination until the is about the major part. Okay, the major part of not major part, major pathways that happen that involve the removing of amino group. But then, as I said, but we also have the minor pathway of this how to remove the amino group from the amino acids. And how does it take place is that okay, so this is the removing of the ammonia from the amino group from the amino acids. All right, so we have transamination, we have the oxidative deamination, and also in the presence of glutamate dehydrogenase under the oxidative deamination, and we have the oxidative deamination takes place in the mitochondria, and it will give up the ammonia or amino group from the glutamate via deamination. So the ammonia will be released. Okay. So this is the process that takes place in the major pathway that we have discussed before. But then this is when we talk about the minor pathway, so there are a few amino acids that involve for the removing of the amino group from in a minor pathway. Okay, which we go through this what we call as direct deaminations. So the first amino acids that actually can remove the amino group directly, which is from in the process of a direct deamination, is the serine. Okay, serine. So this is the structure of the serine. So this is the amino group and H3 here. So what happened is in the presence of the what we call as a serine dehydratase. Okay, so in the presence of serine dehydratase, so the enzymes or the catalyzed so this serine dehydratase will convert the serine into amino acrylate, right? So of course, in the presence of with the removing of the water, okay, and then we have a form of amino acrylate here after the serine, and then next amino acrylate will be converted into pyruvate. Okay, it will be converted into pyruvate in the presence of the water, and then this amino acrylate will remove its amino group to the ammonia here, and we got the pyruvate. Okay, so that is the process how the direct deaminations that happen from serine into pyruvate. So meaning that here. Yeah, so a minor pathway that involved in the amino acids, it actually doesn't go through the direct process of transamination and oxidative deaminations, right? So here, the process of amino group, the removing of the amino group from these amino acids, it happened in a shortcut, okay, like a shortcut, a short way, short pathway. So where the amino group is removed, and at the same time also it also produce or it's also remove the part of the amino group as the ammonia. Okay, and we get the a form of keto acids as well during the process of direct deaminations that involve these amino acids. All right. So as you can see here, we have the serine, and then it's remove the amino group from the main carbon, okay, main skeletons of the serine, and then we get the ammonia, which is ammonia is also released in this case, and we also get the alpha keto acid, which in the form of the pyruvate, right? So this is the for in case of the serine, and other than that, we have also a process of the amide hydrolysis, which we call as amide hydrolysis. Okay, amide hydrolysis is where it can actually directly remove the amino acids from the asparagate and glutamines. Okay, so in the presence of the particular amino acid, it will be converted into pyruvate. Okay, so this is the process how the direct deamination that happen from the amino group to the ammonia here, and we got the pyruvate. Right, so this is the the process how the enzymes, is asparaginase and glutaminase for the glutamine, and they can actually will be converted into the aspartate and glutamate, and also in the and also the ammonia as the product. So this is the for asparagine and glutamine, where these two also can go through what we call as minor pathway or minor or direct deaminations from this direct removing of the amino group, which is go through this amide hydrolysis. Okay, right. And okay, so yeah, so the in case of the when at the end of the oxidative deamination, as you can see, we have the both of the process, whether it is a minor pathway or major pathway that involve the transamination, oxidative deamination. So both actually the products will be the releasing of the ammonia, right? So what happened is that ammonia is actually toxic and it can cause death, and also of course it will actually jeopardize the nervous system, and therefore to detoxify such ammonia, which is toxic. So how is going to be removed from the body? Okay, whether human, whether animals, whether birds, whether bacteria. So actually, they have a different mechanism on how to remove this ammonia from the um, uh, from the blood. Let's say in human or animals, right? So in this case, the ammonia will be converted into urea, okay, which is the water soluble. So urea is actually a form of water soluble, and it is readily excreted by the kidneys in urine. And okay, last week. So last week from the previous from our practical, lab practical. So we actually what we did was we measure the activity of the urease, right? Urease, which is the enzymes that responsible to catalyze the hydrolysis to hydrolyze the urea into the ammonia, right? um, so um, so so that what we did last week, right? So here, try to relate to what the information that perhaps when you figure out, when you try to um, uh, prepare your lab report. So you already read about all these urease or urea, and yeah, try to relate to these informations that you learn in this topic, right? So so after the ammonia or amino group, after the amino group is being released as the ammonia, and also it will be excreted in a form of urea, which is a soluble form of a water soluble form of the ammonia. So the remainder of the amino acid is what we call as carbon skeleton, right? So because we already removed the part of the amino group from the amino acid, so what's left is that the carbon skeleton. So the carbon skeleton of the amino acids, it will be degraded, or it will be processed in a different pathway after that. Okay, after we remove the amino group. Okay. Okay, so here the ammonia, which is toxic, okay, will be formed by the action of the transaminase and the glutamate dehydrogenase from the process of transamination and oxidative deaminations, and what's and the process after that is how to remove this ammonia, which is in a high concentration. Okay, so because this is very toxic. And what happens is that the ammonia will be channeled into what we call as the urea cycle to remove this part. Okay, urea in a form of, because urea is a soluble form of the ammonia. Okay, so urea will be excreted in the kidney, okay, in a form of urine. Okay, so this is ammonia. So we have, as if you will notice that ammonia is in not ionic form. Okay, ionic form is NH4 plus, but in the form of molecule itself, ammonia is NH3. Okay, NH3. So we have N here, and the blue one is N, and we have 3 hydrogen which is attached to the nitrogen. And how is N transported in the blood? Okay, so N, or this uh, uh, or we call as the source of the nitrogen. Okay, the source of the nitrogen compound is actually transported as the glutamine and alanine, yeah, in the blood. Okay, so that's why we we'll be talking about this what we call as the apa ni? Glucose alanine cycle last time. Okay, I already mentioned about glucose alanine cycle, which is because this is where how we want to relate, okay, the contribution of this amino acid degradations or formations of the amino group, or formation of the ammonia to this glucose alanine cycle. Okay. Okay, 2 mechanisms to transport the ammonia, okay, and for the compound that contains the nitrogens, okay, from the peripheral tissues to the liver. So the first one is the glutamine synthetase. So glutamine synthetase is actually will carry, will channel all these nitrogen compound, okay, from most tissues to the liver. Okay, and then in case of the glucose alanine cycle, is the cycle where these play a role to transport these ammonia or whatsoever the nitrogen containing compound to the liver from the muscle. Okay, so that is the two important parts. Okay, which actually play a role to transport the nitrogen containing compound from the most tissues and also from the muscles. Okay, so any questions so far? Any questions? So far clear. Okay. Right. So if no questions so far, then I will continue. Okay. Okay, so in case of the glutamine synthetase, okay, glutamine synthetase is actually the enzymes that involve to transport the nitrogen containing compound to the liver, okay, from most tissues. So here glutamine is actually not. This is a non-toxic, okay, non-toxic form of the ammonia. Okay, glutamine is not. This is a non-toxic. But then in combinations of the two enzymes, we have here glutamine synthetase and also the glutaminase. So at first, this ammonia is not, and actually is not, this is not a form of, this is non-toxic form, okay, of the ammonia. So when it become a glutamine. So we have here the glutamate. So glutamate, because we have a very high concentration of the glutamate when all the amino acid is going to be, when all these amino acids converted into the glutamate, right, after the transamination process. And also oxidative deamination. So we get a very high concentration of the glutamate. So after that, what happened is that the glutamate here will be converted into the glutamate, okay, before it will be transported to the liver. So in this case, we need the presence of the glutamine synthase, and then it will be converted into the glutamine. Then the glutamine will be transported to the liver, because the glutamine will be it can be absorbed in the blood. Okay, so it will be transported to the liver. So in this case, the conversion of the glutamate into glutamate, it needs the requires the ATP, and then the amount of the enzymes increases when the muscle catabolism is occurring. So meaning that the amount of the enzymes in case of glutamine synthetase and also the glutaminase is going to be increased. Yeah. So when this catabolism takes place. Okay. So we have the presence of the glutaminase in this case when we want to convert back the glutamine into this glutamate, and then glutamate will go through this process, so which involve the glutamate dehydrogenase. So what is the process that involved here? This part. What is the process here? All right, good. Yeah, so oxidative deamination. Yeah, um, so this is the part where the oxidative deamination takes place. Okay, after we get after the glutamine, which will be transported to the liver, and we have the glutaminase, which is convert back the glutamine into glutamate, and glutamate will go through the oxidative deaminations to get back the alpha ketoglutarate, and we have a form of ammonia, and then ammonia will be converted into the urea. Okay, so here, meaning that the NH3 here, the ammonia is it's not is non-toxic form in this stage. Okay, it is a non-toxic form, but then this is where the it will be processed. Okay, the glutamate will use this NH3 to convert into the glutamine. Okay, so so it will actually, this NH3 actually will be carried out from the smooth tissue in a form of glutamines to the liver. So here, when the glutamines reach the liver, then this glutaminase, okay, will actually change back to the glutamate, and we have the form of this ammonia, okay, back to this liver. Okay, so we have one in the tissue, and then it will be transported to the liver by using the glutamine. Okay, so glutamine, we can say it's like a transportation to actually bring this ammonia NH3 to the liver, and after get here, then the glutamine will be converted to the glutamate, and we get back the ammonia. Then ammonia will be released as the urea. Okay, here in the the process takes place. So this is very important because we cannot actually have the ammonia directly into the blood. Okay, we don't have such kind of things where ammonia can actually be absorbed in the blood directly. So in that case, the enzyme will change the glutamate into the glutamine, because glutamine can be transported in the amino acid structure of the glutamate. We have the NH3 here, and then in the presence of the glutamine synthetase here, so then the ammonia, the NH3 will be applied, or will be attached to this the end of the carboxyl group of the glutamate. Then we get the glutamine, and this is where in this form where this ammonia will be converted or be transported to the liver in a form of glutamine. Another example for see the how so this is during the movement of our muscle. So the muscle needs actually they need the energy, right? They need energy, and also during the process to form the energy, okay, to form the energy because we have the glucose here, and we go through the process of glycolysis to produce the pyruvate, because the pyruvate will go through the process what we call as gluconeogenesis to actually produce the ATP, right? So, but then in this process, how to get this pyruvate is actually comes from the glucose, right? So in this case, when we get the pyruvate, in this case, the pyruvate, other than it will channel into the process what we call as gluconeogenesis and so on to provide energy. But then the pyruvate also will be degraded in terms of in the process of the uh, transaminations, okay, and we get the alanine, okay, we get the alanine alpha ketoglutarate, and then this alanine, okay, it will be transported to the liver through the blood channel, as I said, because the ammonia it can be carried out to the liver by transporting this ammonia in a form of glutamine and alanine. Okay, so this is why the we get the alanine, and alanine will be channeled into the blood channel, and then we have the alanine to the liver, okay, in a form of the alanine, and then we go, and then alanine will go through this process of transamination. Then we get back the glutamate. Okay, the glutamate then will go through the process of the urea cycle, and we get the urea, and the pyruvate will go through this process of gluconeogenesis back again, and we get the glucose, and then glucose enter the blood, and the blood will channel the ATP in the form of glucose to the pyruvate, and our muscle get back the energy. So this is the cycle, what we call as glucose alanine cycle. So this is a very important, okay, very important part to actually why we call the why we say that the dietary protein also can provide or can contribute to the energy. So this is why, this is how the thing is. Okay, so because we know that okay, carbohydrate the main or major contribution to energy. Yes, that is true, because the carbohydrate is the short the pathway is a little bit shorter, right, compared to the protein. So that's why we call the carbohydrate as the major product, major contribution to the ATP or energy, because it can it can contribute to the energy in a short time compared to the protein, because the pathway is shorter. But then when we talk about the protein, how the protein also can provide to this contribution to the ATP or energy. So this is the pathway that involved. Okay, so the degradation of the dietary protein, it will go through this process to pyruvate to alanine in case of glucose alanine cycle. Okay, so um, so here okay, we have one part here where the glutamate will be uh, uh, converted to this alanine. All right, so here in the process in the muscles, and this is actually the process where the anaerobic conditions only. Okay, so under anaerobic condition, so this actually just happen or occur just in a condition of anaerobic. Okay, and so in case of where um, the muscle contracting rigorously, so Can you understand this statement? So when the muscles, okay, when we so let's say you are out of energy, okay, maybe let's say you are running or you are out of energy, so no more glucose uh, that can be processed in your energy in the body. So this is where the last results, okay, the last results where this um, this glucose alanine cycle will be working. Okay, so we have the so in this process will be involved. Okay, will be involved under anaerobic condition. Okay, so this is glucose alanine cycle. Okay, glucose alanine cycle. We have the okay. So this is just another okay, so this is another figures or another summary to the process of glucose alanine cycle. So this is actually the same things that's described here. So here is just that. So we have the liver in this part. Okay, so this is the same takes place. So we have liver, and we have the muscles. So how the glucose get back to the blood, and then glucose to this transported by the blood channel and go back to the muscle, and the glucose then will be converted into the pyruvate, and then we have the pyruvate is the through the transamination, then we get back the alanine, and alanine. But then here in the process, of course, the pyruvate will go through some process of the gluconeogenesis and so on to provide the ATP to the muscle. But it's to mention here, we don't have such kind of pathway uh, 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 in its case. Okay, but then so here we have the alanine. Okay, we have the alanine and get back to this the blood channel, and then alanine will go through the process of the uh, transamination. Then we get back the glutamate, and this is where um, um, the ammonia, okay, the amino group that we get from all over from this all branching amino acid uh, will be carried out through this alanine, and we get back the glutamate, and this the ammonia here will be aspirated out as the urea. Okay, and so so all right. So this is just the summary. Okay, how the glutamine will be converted back to glutamate and the ammonia in the liver, and then in the liver as well, the ammonia will be converted to urea. Okay, so this is the form of the urea. Okay, so this is the structure of the urea. Okay, so this is the summary of the amino acid catabolism, amino group catabolisms, how the amino group just the amino group, okay, because how the amino group itself will be we go through the process starting from the removing of this amino group from the amino acids. Then we have the part of the transaminations here, and then yeah, so we get the in a form of NH4. So we, as you can see, we have the amino group in a form of NH3 plus, NH3 plus, and then towards to this way to the removing of the ammonia, if we get here NH4. Okay, so this is the soluble form of the ammonia. Okay, so this is also the NH3, the amino group from the alanine, which comes from muscles, and then this is the NH3, which actually carry out the nitrogen-containing compound or amino group in a form of glutamine, which actually comes from the most of the other tissues, okay, other than the muscles, but also muscle also can be contributed to the formation of the amino group in a form of the glutamine. Okay, and uh, and both both sides, whether it is NH2 or NH3, will be converted into the ammonia before it will be converted into the urea. Okay, so this is how the amino group catabolism for the amino acids. And in case of the other animals, so let's say we have the ammonotelic animals, let's say fishes or amphibia, they have a different mechanism or um, uh, different way how to actually excrete out the ammonia, okay, from the system. Okay, and the same thing goes to the urotelic animals, for example, terrestrial vertebrates like birds and also sharks. So they have this mechanism that we call as urotelic, where they produce the uric acid, yeah, so to actually remove the ammonia from the system. Okay, so all right, so okay, so that's it for the amino group catabolism. So the next one, we're going to look at the urea cycle itself, because we know that the ammonia is going to be processed into the urea, which is this is where the ammonia actually enter the urea cycle, okay, to convert this ammonia to the urea, and we're going to look at this urea cycle. Okay, we are going to focus in urea cycle later to see what is actually happened to such ammonia. Why does the ammonia can actually convert to this in the form of the urea? Okay, so we're going to look at the urea cycle. Okay, we're going to take 5 sekarang pukul 10. Okay, so we're going to take 5 lah. Okay, and we get back to the urea cycle later. Okay, so if you have any question, you can put anything. You can put your question in the chat box, and I will look at later after the break. Okay. Okay, let's continue with the topic. This is part two lah. Okay, part two of amino acid catabolism. So basically, we're going to cover on the urea cycle, and then a few other topics which is related to the urea cycle, right? Okay. So everybody still there?
Yes, doctor.
Okay, can you hear me clearly? Because I think my voice macam post COVID, and then now it's fasting month, so basically I cannot drink the water, so tekan macam cepat kering sikit. So so then okay, let's continue with amino acid catabolism. So the part two. Okay. Okay, so before that, I would like to introduce to you with the summer program. Okay, summer program from Kanagawa University, which is now open for applications. If any one of you interested to join this Kanagawa University short-term or summer program, please apply before April 22nd. Okay, before 22nd. If you any of you interested to apply, then please text me later after class, right? And there's just 3 weeks, and you can actually you can mingle around with the Japanese and also other students from different country, doing all these activities that related to the culture, history, so all those things about the Japanese culture, right? So if any one of you interested, please text me after the class. And this is where the Kanagawa University is located. So this is near the sea here, and this is the university, and this is the location of the university. So quite near to the sea, right? So very interesting. So please take this chance. And this program will be carried out from July 19th until August 9th, okay, for 3 weeks. And you can actually apply for this is limited for first 25 applicants. Okay, scholarship is just available for 3 selected candidates. You can apply the scholarships to actually join these activities from the IPTRA, and it's just limited to just first 25 applicants. But the scholarship is just just 3 selected candidates, right? Okay. Okay. So let's continue with the next topic about the deamination. Okay. So talk about the amination just now from the previous slides. So the amination is a process where we actually remove the part of the amino group, right, from the carbon skeleton of the amino acids. So when we remove this amino group from in the process of transamination and oxidative deamination, this is what we get is just the carbon skeletons of the amino acids. All right. So the part of amino group that has been removed from the carbon skeleton is the amino group, the amino acids, or the amino group will be excreted as the urea. Okay, so it will go through the normal pathway if, let's say, the normal enzyme presents to in the urea to process the urea. But then, if, let's say, there is no such the normal pathway presents or normal enzyme presents in the process, then the urea will be the ammonia will be built up to the toxic levels, and this is what we try to get rid of. So that's why I want to make sure that the kidney can so your kidney is healthy, and so on. So this is where the process of the urea will take place in the kidney, okay, in the kidney, in the liver. So that is the location, is the the important organ that uh, play a role to process all this ammonia, which is highly toxic. All right. Okay, so I'm not sure if you noticed that in the old people, basically, if you notice your grandma or your grandpa, or if you notice they have this kind of spot in the skin, yeah, in the skin. So basically, this is the actually the indicator that they have the problem with the liver. Okay, so mungkin you can check your own hands or your own skin as well. If you find out you have this kind of spots in your hands, in your hands or other parts of your body, then most probably you have this kind of you have this problem with the liver. Okay, so this is what we call as cirrhosis. Yes, cirrhosis. So cirrhosis, where the liver becomes yeah, this kind, they have this kind of formation, yeah, of lump in the liver. So this is normal liver, but this is the liver with cirrhosis, where the problem become, where the liver become abnormal. Okay. Okay, so in this case of the urea cycle, so urea cycle is where the process takes place. Either the cycle that will do the process of to process this urea from the ammonia to the urea. So we know that the ammonium salts, okay, from the oxidative deamination. So remember, after oxidative deaminations, we have the product, which is the alpha glutarate and also the glutamate. And then the glutamate, the part of the amino group of the glutamate will be degraded, right? And we have this high concentration of the ammonium, of course, in the blood. So we don't want this ammonium salts to increase until toxic levels, right? So if, let's say, the ammonium is present in a very high concentration, which exceeds the 0.25 millimolar, all right, so in the blood, so it can cause the vomiting and coma and even death, right? So this is a very important, yeah. So you have to make sure that the concentrations of the ammonia salts in the blood is less than 0.5. And so therefore, we know that it's very important for us to get rid of this ammonia from the body, right? So in case of the other organisms as well, they have a different mechanism on how to get rid of this ammonia, high concentration of ammonia. So for example, we have the fish ammonia. Okay, fish or in fish, they get rid of this ammonia through the gills, which is from the mechanism what we call as ammonotalism. So this in the fish, and in reptiles and birds, they excrete mainly in a form of uric acid, and this is from this pathway what we call uricotalism. And for mammals, we excreted it in a form of urea, so we call it ureotelism. So this is the different pathway that takes place in the different organisms just to get rid of these ammonium salts. All right. So in mammals, we synthesize the urea through this process of what we call as ureogenesis, that which is occurs in the liver. So this is where the location of the urea will be processed. Okay, we will synthesis, which is we will synthesis from the ammonia, and the urea is the product from the urea cycle. Okay, so this pathway of the urea cycle was actually found by the Hans Kraft, and this scientist occurred during 1932. So these two scientists, they figure out about this, or they actually elucidated these urea cycles where it play a role to synthesize the urea out of the ammonia. Okay, so this is the structure of the ammonia, of the urea. Okay, of the urea, where you can see this. There is the two molecules of NH2. Okay, at both sides of the ammonia of the molecule, right? And okay, so the main thing about the urea cycle is that it consists of four reactions. Okay, this is the urea cycle. It consists of four reactions, and the first reactions occur in the mitochondria. Okay, so in the mitochondrial cells in the liver. Okay, and then the other three reactions occurs in the cytosols. Okay, so there are two. We can say they have two parts. Okay, two parts, which is the first reactions happen in the mitochondria, and the other three stages happen in the cytosol. And in this case, to actually to channel whatever products that comes from the mitochondria to the cytosols, they require these two transport proteins, which will connect whatever products from the mitochondria to the cytosols matrix. And this is what we, for the cells, required, which is the citrulline ornithine exchanger and glutamate aspartate translocase. So these are two transport proteins, very important to channel, to link whatever reaction happened in the mitochondria and also the cytosol. All right, so so if you can see here, this is the first stage of the urea cycles happen in the mitochondria, where they actually prepare for the carboxyl phosphate. Okay, they prepare for the formation of the carboxyl phosphates in the presence of carboxyl phosphate synthesis 1. So what we need to, or what actually the needed to form this carboxyl phosphate, because carboxyl phosphate is very important to actually transform or to be converted into this citrulline. All right. So what we need, what the mitochondria needed for the carboxyl phosphate is that they need the ATP, and then they need the hydrogen carbonate and the ammonia itself. Okay, so they need the ammonia, and then they need the hydrogen carbonate and 2 ATP. So of course, in the mitochondria, the process will involve the energy, which is equal to ATP, right? And then and they need the presence of this enzyme, what we call as carboxyl phosphate synthesis 1. So this is very important to convert this hydrogen carbonate and ammonia to the carboxyl phosphate. And once we get the carboxyl phosphate in the mitochondria, then this is where the proteins that will be transport this carboxyl phosphate to this to the formation of the citrulline and enter the cytosol. Okay, cytosol. And we get the citrulline, and this is we have the three steps. Okay, three steps or three major steps that happen, that occur in the cytosols. So, which is the formations of the aspartate, and then formation of the fumarate, and then formation of the urea. So that this is the three major steps that happen in the cytosol. Okay. So the first thing is the citrulline will be converted into arginine succinate, and in this case, the aspartate will be produced. And the second part is the arginine succinate will be converted into arginine. Okay, and of course in the presence of the arginine succinate layers, and we have the fumarate as the product, and finally the arginine will be converted into this ornithine. Okay, ornithine. So the product will be the urea, and they need the enzyme, what we call as arginase in this case. Okay, so that is the process. another three major steps that happen in the cytosols for the urea cycle. Okay. Okay, so this is the about carbamide phosphate. So carbamide phosphate that produced in these locations in this mitochondria is actually a very unstable high energy compound, meaning that once this carbamide phosphate is produced or formed after in this location in this mitochondria, they need to be transformed into the citrulline in a very in a very short time. Okay, because it's a very unstable energy compound. So this carbon phosphate will be transported to this cytosol in the form of citrulline. Okay, and so this formation of the carbon phosphate takes place in the mitochondria, and they need this presence of the carbon phosphate synthesis one for the process takes place. Okay, so here we need the carbon dioxide. So so if you can see here, we have this carbon dioxide, and will be converted into carbon phosphate. But then here we have, so where actually the source of carbon dioxide in these equations here? Where does this carbon dioxide? Because what I showed you here, okay, so this we need carbon dioxide, right, for us to form this carbamide phosphate. Okay, carbamide phosphate, which is the phosphate comes from this ATP and this carbon dioxide here, forming this carbonyl group of which is attached to the NH2, because NH2 comes from the ammonia. But then we have the CO2 here. But in this equations showing here, where actually the carbon dioxide comes from which source? Do you see any carbon dioxide in this equation? All right, so yeah, that's correct. Yes, HCO3, which is the hydrogen carbonate. So this is where the hydrogen carbonate actually the source of the carbon dioxide itself. So it's stated here HCO3, but the HCO3 is actually a source of the carbon dioxide. So this will form the carbamide phosphate. You can see from the sebenarnya for you to understand further how the carbamide phosphate is formed from these three components, right? From these three components, actually, you can see it from this structure itself. So if you can see from the carbamide phosphate structure, so this is the phosphate. The phosphate is, of course, comes from the ATP itself, which is the ATP will be degraded into this 2-ADP, and at the same time, it will produce the phosphate. So the phosphate will be attached, okay, to this CO, which is come from the carbon dioxide, and also from NH2, which comes from the NH3, from the ammonia. Then they merge together all these components, all these elements, and they become the carbamide phosphate, right? And the next one is the formation of the citrulline. Okay, the citrulline. So what interesting is that during the process of expressions towards from the mitochondria to the cytosol is that where the ornithine, okay, or the ornithine. So so this is the ornithine. So the ornithine is the basic component or the basic element in the urea cycle, actually, because whatever the last product that will be produced in the urea cycle, it will be used, or it will be recycled back into the cycle. Okay, it will be recycled, it will be reused into the cycle. So meaning that um, uh, so the basic element, okay, the basic element ornithine, so it will bring this the component of the carbon phosphate to form the citrulline, and then the component, the citrulline will be converted into arginine succinate, to the arginine, and then back to the ornithine again. So meaning that ornithine is like a transport, okay, like a transport to bring all this component. So here we have the ornithine. So if you look at here, we have the structure 1, 2, 3, 4, 5, 5 carbon of the ornithine. It's attached to the NH3 and 2 NH3 at the first carbon and the fourth carbon. And when this carbon phosphate comes in, it's actually remove the part of the phosphate, and then the part of the carbonyl group which attach to the amino group will attach to the first carbon of this ornithine. Okay, so if you can see here, we have the right. So we have this component here. Okay, this component. So this phosphate group will be detached from this carbon phosphate, and we just have this element which is attached to this part. Okay, the first carbon of the ornithine. Okay, so basically we're gonna have just NH. Okay, just like this, and this is the part of the carbonyl group of the carbon phosphate will attach to this first carbon of the ornithine, and they will become this citrulline, and they ready to enter the cytosol. Okay. 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 So the next one is that, so once in the cytosol, the citrulline will condense. Okay, will condense with the aspartate, because the aspartate will comes in. Okay, will comes in, and in the presence of the ATP, the citrulline will be combined with the aspartate to form this what we call as arginosuccinate. Okay, so this is the arginosuccinate. So the part of the aspartate will combine to this location of the first carbon of the ornithines. So the same thing. So meaning that the first carbon of the ornithine is a very important part where it attach the carbonyl group of the carbonyl phosphate, and then the second reactions in the cytosol is that it will condense, or it will attach to this component or element that comes from the aspartate. Yeah. So still, this first carbon, play a role for every single reactions. And then the next one is after. Then we get these arginosuccinate, where the arginosuccinate will be it will then cleave the part of the one that attached to this aspartate just now, and we get the arginine and also fumarate. Okay, and fumarate will be eliminated in the cycle, but the arginine will go through to the next step. Okay, so you can see how this arginosuccinate is cleaved. Okay, where this this part will be cleaved in the presence of the arginosuccinate layers. Okay, so you can see this part is actually removed. Okay, and we get this arginine. Okay, and and here the arginine will be cleaved by the arginase. Okay, so finally, so this arginine here will be cleaved. So this part will be cleaved, and it will regenerate back to the ornithine. Yeah. So we get back again the ornithine as its original form. Okay, after the arginine is cleaved, and this part will be uh, uh, available to the formation of the urea. Okay, so this is urea. And this ornithine will be ready for the next cycle. Okay, so next cycle. All right. Um, so this is the reaction that takes place in the cytoplasm and mitochondria. Okay, so this a little bit how can I say, a little bit detail, okay, a little bit detail, which involves um, the blood channel here, which is the one that not blood channel, the enzymes, the transport proteins, the transport protein that actually link between what the process happen in the mitochondria and the cytoplasm here. So we have when the carbonyl phosphates produced, and it will be in the presence of the ornithine, that which is this ornithine actually comes from the cytoplasm itself, but the ornithine will go to here. Okay, we'll go to the part of the uh, mitochondria. Okay, so as you can see here, the ornithine that produced will go to here to actually accept this carbonyl phosphate, and we get the citrulline as the product. Then the citrulline will be channeled into the cytoplasm. So once in the cytoplasms, then they will go through the three main stages in the urea cycles. Then you will produce this urea. Okay, so this is the main products that the main products that should be in the urea cycle. And we have also the fumarate, and the fumarate, as you can see, as we have discussed on the urea cycle just now, the fumarate also will be eliminated from the urea cycle. So where does this fumarate will go? So it will go to this TCA cycle. All right, so TCA cycle. And the CA cycle will change, will convert fumarate to malate and the oxalic acetate. And this is oxalic acetate. Remember, we have the oxalic acetate in the process of transamination just now, right? So the transamination of oxalic acetate will be converted back to aspartate, and the aspartate is very important because aspartate is one of the substrate that needed in the process of urea cycle, where the citrulline need to be condensed with aspartate to form the arginosuccinate just now, isn't it? So this is where the a little bit detail, which is involve the process of transamination that happen in the cells, in the muscles, in the most tissues, and also how the relationships with the TCA cycles. So this is very good. I think it's a very good figures to give you an overview of what is happening. Not just we focus on the urea cycle itself, but then you can see how you can corroborate this urea cycle and the products with the other pathway as well, right? Okay, so so this is just the informations of the urea formations. So this, as I said, this occurs in the liver, excreted by the kidney, and the principal method for removing ammonia. So we have this one of the process of removing ammonia. So let's say there is a defect. There is a defect in the recycle enzyme. So let's say we have so many enzymes that involved in the urea cycle. So we have arginosuccinate, lyase, and so on. Carbonyl phosphate synthesis 1, and those all the important enzymes that actually play a role to synthesize all these steps in the urea cycle. But then, what happens if there is abnormalities? There is something wrong happened to these enzymes, so that there is no presence of the enzyme. So we have another we need to know another pathway or another steps that can actually can be carried out to remove the ammonia. So the first thing is in the process or in the conditions what we call as hyperammonemia. So this hyperammonemia is the where the ammonia is of course in the high concentrations because of the defects of the urocycle enzymes, and in this case it will cause a severe neurological defects, especially in the in for babies, and so um, and of, and of course in every stage of life. And what is the treatment? So the first treatment is we have to stop protein intake, because we know that the ammonia, the ammonia, the amino group is the um, the main source, right? The protein is the main source for the amino group, or the accumulations of the amino acids. And the next one, what we have to do is to do dialysis, and I think this is quite familiar as well for you guys, because when you know someone ada sakit buah pinggang dan sebagainya, they have to go through this dialysis process. So this is why the dialysis is very important to actually to remove the toxics from the body, especially from the nitrogen containing compound such as ammonia. All right. So in the increased ammonia excretion, we can actually increase ammonia excretion by taking these drugs. Okay, sodium benzoate, sodium phenylbutyrate, L-arginine, and L-citrulline. So these all the compound or the drugs that can be taken to actually increase the ammonia excretion. All right, so this is summary of amino acid metabolism. So we started with the dietary proteins, right? So dietary protein, and then the amino acids in the blood, because after this will be degraded by the enzymes. Then we get the simple amino acids in the blood. And what happened is that this amino acid will be somehow this amino acid will be synthesized into a larger macromolecules like proteins, enzymes, or other components in the pathway. But then the part of the amino groups will be synthesized individually, or other than the carbon skeleton, as you can see, what happened in transaminations where it involves the process of the nitrogen or nitrogen containing compound or amino group. So it will go through this transamination, oxidative deamination, and so on. And it go through this. It will enter the urocycles, and it will be excreted as a urine waste for the ammonia. And for the carbon skeleton, we haven't discussed in this case so far, but it actually has a different pathway how to process the carbon skeletons of the amino acids to actually produce the energy as well. Okay. And this is the this is also a summary of the urocycles. So this is the include the structure formula for each of the components, each of the intermediates that are involved in the process. So starting with the ornithine, so this is the ornithine, where the ornithine will combine, will actually receive the part of the source of the carbon, right? So it will receive the whatever the product from the carbon phosphate, and then as the citrulline in the cytosol. And so this is where the other process, okay, the other intermediate takes place. And you also can see how the formations or the changes, okay, the changes that involve from one intermediate to the next, just to relate of what is happening in each process or in each stages. And there is also provide to you in this figure the enzymes, okay, the enzymes are involved for each of the process. All right, so this is the summary of the reactions that takes place in the urea cycles, together with the what are the enzymes and where is the locations that involved for each of the process. So as we know that the first we have the first reactions. We have one reaction that happened in the mitochondria, and then we have another three which is happened in the cytosols. So this is three that happen in the cytosol, and we have the extra process that happen in the mitochondria, because the ornithine actually go to the mitochondria to receive this the formation of the carbon phosphate. Okay. Okay, for a summary, so what we have learned so far in this topic. So the body by deamination or deamination of amino acids. So this is what it's all about, the deamination. So when we talk the amino acids, so there will be the degradation of the amino acid, which of course the source is from dietary proteins or from other protein that already accumulate in the body itself. So it will be it will go through the process of deaminations, and the process of deaminations starting with the transaminations, and then we have the oxidative deamination, right? And then so this is the transamination takes place, and then we have the major and minor pathway as well. So if you can see in the major pathway that involve the alanine, aspartate, and we have the minor pathway that we have it starts on the serine and glutamine, and also another amino acids that involve in the minor pathway, and all this will actually comes with the source of the ammonia, okay, because they have the part of the amino group which will be removed from the amino acids, and they comes as the source of the ammonia. And what happened is that this ammonia uh, uh, will be channeled into the urea cycle. Okay, all right. Okay, so this as we can see um, from the earlier part, from the first process, from the beginning until the end. So how we can see the glutamate. Okay, the glutamate is a major role in the metabolism of amino acids, where it's involved both degradation and the synthesis of the glutamate. So it's just not that the process of the uh, transamination until the end of it just involves the synthesis of this glutamate, but that is also involved the degradations of the glutamate itself to make sure that everything is balanced from the from the beginning until the end of the process. Okay, so meaning that from the first part, where we get these amino acids, and we get the glutamate from the transamination, and then at the same time also we have the oxidative deaminations back to produce the um, glutamate, and we have a form of the ammonia here, and ammonia also can come from other direction as well. And we have the ammonia, and ammonia will enter the urea cycles, and we already discussed about the how each step of the urea cycle itself, from the beginning and from the first step in the mitochondria, and then we have another three steps in the cytosols, and towards the formation of the urea as the product from the urea cycle. And from there, we also discuss how the relationships of what happened to the side products of the urea cycles to the other pathway, like to the TCA cycle, and also to the transamination, or to the process of formation of the amino acids or degradation amino acids. Right? And another thing to pay attention as well is about the ornithine. Here, the ornithine is a compound that both initiates and is also regenerated during the urea cycle itself. As I said, ornithine is macam the basic element here in the that maintain in the urea cycle. As you can see here, we have the ornithine. So the blue one is the blue one is actually represent the ornithines. So this is the real structure formula for the ornithine. Okay, so which is represented by the blue square like this, not blue square, blue tag, okay, blue tag. Like this, so you can see we have the ornithine right here, and ornithine will receive the carbonyl group from the carbon phosphate, and then it will be condensed with aspartate to produce the arganosuccinate in this location, in this stage, and then arganosuccinate will be converted into arginine with the fumarate will be eliminated in the process, and back to this ornithine where the urea cycle is produced. So you can see this ornithine is like a tulang belakang. It's like tulang belakang to each of the element in the urea cycle. So it's very important. Okay. Okay. So then, what happened to the carbon skeleton for the amino acid? Because amino acids is already being the amino group is already being processed until the urea cycle. So that is the end. That is the end fate for the amino group. But then what happened to the carbon skeletons? Because this is what we haven't we haven't discussed about carbon skeletons. Okay, so so I think we were going to discuss about the what happened to the carbon skeletons, how the carbon skeletons will be processed in the next class, in the next lecture, on the Wednesday. So we have another 1 hours for that. I think it should be enough time for me to finish the topics for the carbon skeletons. How do we, or what's going to happen to the carbon skeleton after we remove the amino group. Okay, so I'm going to end here for today. Okay, any questions? Any questions? Tak ada? Okay, Wednesday we will do it online juga lah. Okay, so the attendance for today, I will put I will upload. I akan buat link dekat pecah belas later. Okay, I haven't do it, but I will do it right after this class. So just go there and just sign your attendance lah from there, from the link. Okay. So another 1 minute. Anything?